Hello, I don't have another class on Fridays.
Oh,
That was the only class I have, so I'm here.
all right. Glad to have you. All right. So the question before we do anything, what do you want to know? What questions do you have?
I'm just trying to sort out the difference between mitosis and meiosis, like what actually happens to the chromosomes versus chromatids. Like I'm just trying to mentally straighten that out.
Okay. Let's start with that then. And we'll work our, somebody else? Did somebody say something? Okay. All right. So when we're talking about, well, you know what? I'm going to see if I can do this on my board. I never get to use my board, but it's easier than writing on the shared screen. So let me see. Let me make sure I've got it on record. And the important things with mitosis and meiosis. One of the things that, first of all, well, let me try this again. First of all, you've got to remember which one is which. Mitosis is for everything. Meiosis is only for a sperm and an egg, to make a sperm and an egg. So that's what you want to remember. So with mitosis, when you get done, you have to have the same thing that you started with. But you have to keep in your mind that what you're starting with is a replicated chromosome. So remember, I know in lab we did this. We had this, right? We had this kind of shaped thing. And that is a single chromosome. Can everybody see that all right?
Yes.
Yes.
But then that is also a single chromosome. But it's been replicated. That's been through S phase. So before S phase, it looked like the first one. And then after S phase, it looks like this. And so when you first see it starting mitosis, you see it already in the form of sister chromatids, right? Those are sister chromatids. And they're identical, right? They are absolutely identical. You have replicated, we talked about replicating DNA, and remember that it's semi-conservative, meaning we took one strand of our DNA and or our double strand, and we split it up and we used one of the strands for a template for a new strand. We use the other one, right? So this is identical to the second one. First one and second one are identical. So that is one chromosome, has all the same characteristics, the eye color, that everything's the same. But we get one of these from, let's say this is from mom, and then we've also gotten the exact same thing. Whoops, not making a very good picture here, but you get the idea, right? And then we've got the exact same thing from dad. And if this is chromosome 6 from mom, its homolog or homologous chromosome is chromosome 6 from dad. So in you right now, you have 2 copies of chromosome 6, one you got from your mom and one you got from your dad. All right. So with mitosis, you want exactly those same chromosomes to end up at the end. But remember, when they end up at the end, they're going to be back to this.
So if I understand correctly, trying to wrap my mind around this, during the DNA replication, it's not that you're doubling chromosomes, because then you'd have double 46. It's that, is it essentially becoming twice as big? Like twice the size?
Yes.
Okay, that's helping my mind process this.
Right. So you're replicating what you have there, but remember through meiosis or mitosis, we're doing mitosis now. Remember that at one point, all of these are going to be lined up at the center, right? So we have all of our chromosomes in the form of sister chromatids lined up at the center. One of those sister chromatids is going that way, and one is going this way. Once that happens, once that separation occurs, and that piece is separated from this piece, now you have all new chromosomes again. And I shouldn't say all new chromosomes, they're the same chromosome, but now remember, this is what we call a chromosome. This is separating, so we only have half of it in one cell, and we have half in the other cell. This is one chromosome again, right? So we went from one chromosome, we divided it, and we now have one chromosome in each cell. Kind of cool, huh?
Yeah, my mind was trying to like process the thought, okay, how do you start with 46 and end with 46, but like you're not doubling chromosomes? It was just a little brain block for a moment.
Yep, understandable. That is a very hard concept to get. And that's why I spend so much time saying, this is a chromosome, a single chromosome, and this is a single chromosome. Because it's hard to get that that's not two chromosomes. Why isn't that two if this is one? So that is a very hard concept to get. So when we do meiosis, we're going to line up together with moms and dads, our homologous chromosomes. They're going to look like this at the start. That's all going to be the same. But when we separate the first time, whoop, that's not going to help. Here. I'm so used to using my mouse for everything. So I'm talking about meiosis now. If this is mom's and this is dad's, and we're going to have crossing over and we're going to have independent assortment. But what we're going to get after one division is mom's going to be in one cell, right? And dad's going to be in the other cell. So we have divided our homologous chromosomes. We still have it in the form of sister chromatids because we've just separated our homologous chromosomes. So we've separated those. So now I have two cells, each with sister chromatids in them. But if this is chromosome 6, this cell has a chromosome 6, and this cell now has a chromosome 6. Pretty cool. The second division, so that is our first division. That's one division. Our second division, I'm going to move it over here because it'll be easier to see, is going to result in two cells. But now they're going to look just like they did after mitosis. They're going to be a single chromosome. They're not going to be in the form of sister chromatids. And that's because one is going to go one way and one is going to go the other when they separate. And this one, this may be dad's, right? But that's going to do exactly the same thing. So that's going to have a chromosome 6 of dad's and another chromosome 6. Remember though that these are going to have variation because they're also going to have some spots that have crossed over, right? So these are all going to look different. Good.
And that's
so.
Four
And that's what?
haploid with the 4 daughter cells.
Yep, each of these is going to be haploid. The cell that has this in it to start, that's going to be diploid, right? Because it has two sets of each chromosome. Remember, this was chromosome 6 of mom and dad. So this is diploid. There's 2 chromosome 6s. But now each of this cell only has 1 of chromosome 6. So that's haploid. Okay, so this is 2N, which is diploid. And this one is 1N, meaning it has only 1 copy of the chromosomes. And so that's called haploid.
Can you explain after that hetero and heterozygous? After what you explaining for meiosis?
Tell me that again. What do you want me to explain?
The heterozygous and yeah.
Heterozygous versus homozygous?
Yes.
Okay, hang on for 1 sec. Are we good with mitosis and meiosis? Are there any questions about that?
I think we're good right now.
Anybody else? All right, let's move to Yokari's question. And if that brings up anything else, ask along the way, okay? And then I'll move, when nobody has questions, I'll move to practicing Punnett squares.
I know for the, what is it, the mitosis, I always mix them up. Prophase, metaphase, anaphase, telophase, cytokinesis. And then for the mitosis, I think it's prophase 1. Is that the right?
No, meiosis. Meiosis is the one that has 2 cell divisions. And with 2 cell divisions, for each cell division, you have numbers 1 and 2.
Oh.
Mitosis is the first ones you said. Remember, mitosis is going to be interphase first, right? And G1, S, and G2. And then it's going to be prophase, metaphase, anaphase, telophase, and cytokinesis. And with meiosis, you're going to have interphase. You're going to have G1, S, and G2. And then you'll have prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis. And then you're going to have interphase again, but you're not going to have S phase. So you're only going to have G1 and G2 in between the stages of meiosis. You're not going to replicate the DNA, all right? And then you go to prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. And since those are all related, you start out going from 1 cell to 2 cells, and then each of those cells goes from 1 cell to 2 cells. So from the original cell you had, your parent cell, you now have 4 cells. 2 divisions, 4 cells in meiosis.
Okay.
Good?
Thanks for that,
Yeah.
because I thought there wasn't a second interface, so that answered that.
There is a second interphase, but they actually call it interkinesis in your book. It's the only place I've ever seen it called that, but it's a perfect word for it. It's in between, but we don't do the whole thing. We don't do the S phase because that's when
That's already happened.
right.
Okay.
Great. All right. Are we good with that? And Yokara, you wanted to know about heterozygous versus homozygous?
Yeah, I got it really messed up on the test for the mastering.
Okay. So remember that you have, actually I should have left part of this, right? You have 2 chromosomes. All right, and because your cells are diploid. And this chromosome is from your dad.
Out of the French.
And that chromosome is from your mom.
That's
And that right there is, hey, girls. My little whiny girls.
It's like the kids.
Yeah, I know. this gene is the same, this trait on both. And this is, let's say, we'll make that unattached earlobes. Okay, we've heard of that. So there's our unattached earlobes. That's where that's located. Let's say that mom has the gene for unattached earlobes, but dad's gene, same spot, has the gene for attached earlobes. These are called alleles. They're matching, they're in the same gene, right? These are homologous chromosomes, because this is chromosome 6 again. I don't know why 6 is always my favorite, but chromosome 6, dad's, chromosome 6 of mom's. This is in you now, you'll carry. You carry both of those, and these are alleles. They're the same gene, they're at the same spot on the same number chromosome. But what you have is you have two different copies, one from mom, one from dad. Those copies can be like this, where dad gave you attached earlobes and mom gave you unattached, or they could be the same. You could have gotten attached earlobes from both dad and from mom at that particular site. If you got attached from both dad and mom, you got the same gene, same allele, that is homozygous. Homo means the same. So if you got attached, you got the same allele from both mom and dad, that's homozygous. But if you got what we had here first, and you had attached from dad and unattached from mom, they're not the same. Hetero means not the same. So that is heterozygous. So to make it a little easier, let's say, I think unattached earlobes is dominant. So this is a big U, and this is going to be a little u.
I think that's where I got it messed up, because when do you know which one is dominant and the other one recessive?
I will always tell you in a problem what's dominant and what's recessive.
Oh, okay.
Read the problem and it will tell you what is dominant and what is recessive. I don't fool you. I don't try to pull anything on you. It's going to say exactly what is dominant, and it may not say what is recessive, but it will say unattached earlobes is dominant and mom has attached earlobes. So you know that the attached earlobe is recessive. Okay.
Okay.
So,
okay.
but let's go back and make sure if we're unattached and attached, then we're not homozygous, right? We're heterozygous, because we're going to have a big U and a little u. So that's how you can tell when you're looking at alleles, they're going to be different. If they're different, then that's going to be heterozygous. If we go back and mom is attached again, then she's also going to have a little u, and that's going to be little u, little u. That is homozygous because they're the same.
I got it.
Okay,
Thank you.
you're welcome.
Speaking
All
of alleles, would we be getting into the multiple trait kind of? Okay.
No, the only thing we touch on with that is the polygenic. And I may ask you what a polygenic trait is, that more than one allele contributes to it, but I will not ask you to do it or to figure anything out from it.
Yeah, those are pretty big. Trying
Yeah.
to do those fun squares on the homework.
Yeah.
Is there like an easier way for you to like tell me dominant, like the meaning I read it, but it's just like I can't understand the meaning they gave me for dominant and recessive. Is there like a shortened form?
Dominant and recessive are short. If a dominant allele is present, it will be expressed. It means it will show up in the phenotype. Phenotype is what the appearance is versus genotype is what the genes look like. So when you're talking about a dominant and recessive relationship, let's go back. Let's look at, let's make mom unattached again. And this is dominant, meaning that whenever you see that big U, whether it is a big U, or whether it's like this in the heterozygous form, it will show up. So if this was the genotype, the phenotype is going to be unattached. If this is the genotype, the phenotype is going to be unattached. If this, remember this is little u, if this is the genotype, then the phenotype is going to be attached. When you have this combination, obviously it's easy to understand this. If you have two of those, it's easy to understand that you have unattached. But if you have a big one and a little one, then it is going to, this is dominant, and I'm telling you that in the problem, so you'll know which one is dominant. But this is dominant, doesn't matter what that second allele is, right? It can be a big U or it can be a little u, and you will see this trait. That's what a dominant trait is. No matter what the dominant trait is paired with, it will show up in a phenotype. Neither of these are dominant, right? There's no dominant allele there, and that's the only way we see a recessive phenotype.
Got it. Thank you.
You're welcome.
How many questions would we be asked on the test about the specific disorders and diseases?
I usually cover them, but I don't get very specific about any of them. I can't really I'm trying to think. I have plenty of information without much on the diseases. So I would probably pick out something from each section and ask a basic question about it. So I would pick out something like CF, cystic fibrosis, maybe Huntington's. I'm trying to think, we really don't cover much in the way of diseases though through this, do we? What other diseases did we covered? Tay-Sachs, we covered PKU. Oh, we did things like hemophilia. Usually, well, and I don't make guarantees. I never know until I actually get the exam done. But many of those types of things would come up in a Punnett square. So I might say a parent of the autosomal who is a carrier of cystic fibrosis, which is an autosomal recessive disorder, has a child with a person who does not. So that would be the most likely way you'd see any of these, but that doesn't mean I might not say something about, I don't know, something basic about one of the diseases. I don't know. I know I'm bad about saying that. I just don't want to say I won't cover something and then have it be on there. Oh, you're right, Taylor, or Tyler, sorry. we did talk about you are absolutely right. I forgot we did the chromosome number and structure. So we did talk about leukemia, and we talked about Down syndrome and the X and Y disorders. I guess we did do quite a bit, didn't we? I try not to get too specific, but I do ask questions about those.
So like if we understood, say, that Down syndrome, comes from, the extra sex chromosome, that might be in-depth enough knowledge for the exam without knowing the ins and outs of what exactly Down syndrome is.
Yes, I would agree
Okay.
with that, but Downs is not an actual, it's not X chromosome or Y chromosome related, right? That is trisomy, and it's actually an autosome. It's
Okay.
number 21, 3 copies of number 21. But things
I'm
like
heard of that.
that chart that had the occurrences, incidents, something like that I would not cover. Usually if I do cover it, I'll give you a clue. If I cover something, it's usually something I've talked about more specifically, not necessarily just the details on that, but I talked about an experience or a story or gotten a little more in depth than just what's on the slide. So I may ask what's on the slide, but it should kind of clue you in that, oh, well, she talked about that. So it's something that interests me or that I have some knowledge of more than the usual.
Okay, that helps.
All right, you want to do some Punnett squares, or do we have questions before that?
Can we do something about the type of blood A, B, O, something like that?
Yep.
Yeah.
How about if we do a codominance Punnett square? And whenever I do a codominant Punnett square, I do it with blood type. So let's do a codominant Punnett square. Now remember, what are the different types of inheritance that we talked about? One was codominance, right? We have the dominant and recessive relationship. We have codominance. Anybody remember the others?
Incomplete, that's the blend, right?
Incomplete, that's a blend. Yep. I have a list of them, and I think we should be able to come up with 2 more. How about sex-linked inheritance? Right? So that's 4. And then the last one was polygenic. And I was telling Jen, I won't ask a problem. I may ask you what polygenic means or what type of trait. We talked about height actually for polygenic, but skin color is another, skin tone is another polygenic trait. Okay, it's why I'm as pale as can be and somebody else has darker, more olive skin or darker skin than that. That is a polygenic trait. So a bunch of different alleles contribute. It's not just 2 alleles, dominant or recessive or anything like that. Okay, so those are our types of inheritance. So let's look at codominance. So I'm going to make my square here. Can everybody see that all right?
Yes.
Yes.
All right. So I'm going to have a mom who is type O. And I'm going to have a dad that is heterozygous, but is type A. Heterozygous and is type A. That means that dad has 2 different alleles, right? But one of them at least has to be A. That means dad can, if it's heterozygous, then he has to have one A allele, one big A, and one I. Mom, because she's type O, has little I, little I. Do you remember this, Yokari?
Yes.
Okay. So when we're talking about codominance and when we're talking about blood type, we use these I's, little I, and then big I. Big I is going to indicate that anything that's got a big I is codominant, meaning they're both represented. So mom,
Does it matter how we place it on the square?
nope,
Okay.
nope. I find everybody does it a little differently. And even if you look in books, there's probably somewhere when we started Punnett squares that you had to do it a certain way, but I have no concern either way. So our Punnett square, IA little I for dad, because he is heterozygous. And then mom is little I, little I. That gives me 1, 2 that are, what blood type are these?
A.
Type A. And what
Is
are
O.
these?
O.
Oh, what's the difference when, I'm going to take that off and follow Yokari's question just a little bit.
How do you know to fill it in like half though? Like with the, that's where I'm messing up. Like 2 eyes, wait, 2 little eyes in each box, and then.
Okay. All right, hang on. We'll do more of those.
Okay.
So if something is little I, remember that when we're talking about codominance, actually, let's skip the little I first then. Blood type A has a specific substance on the surface. So our cell has all these little things on the surface. A is what puts those little things. Remember, A is a gene, right? And so A has the ability to be transcribed and translated into protein. It's actually translated into an enzyme, and the enzyme sticks that substance on the surface. That's its job. It sticks that substance on the surface. B also is a gene. It's transcribed. It makes a protein. That protein is an enzyme, and it puts a different substance on the surface. But if I were to inherit on one of my alleles an A and on the other allele a B, both of those genes are made, right? Both of those genes are going to be transcribed. I'm going to have the enzyme for A, and I'm going to have the enzyme for B. That means that if I get an A and a B, I'm going to have both of these on the surface because I've got the enzymes to make both of them. So my AB are both going to be expressed on the surface. That's called codominance. They go with each other. So they're working together. They're going to make my cell look different than either the B by itself or the A by itself. That's codominance. The catch with codominance, and this would be so cool if it stopped here, but it doesn't. And our little I doesn't get any of these enzymes because they don't get an A or a B allele. So they don't get any of these enzymes. So a type O can't put anything on the end because it doesn't have an enzyme to put this either little ball or this little triangle thing. Okay, so my I doesn't get any of that. This is where it gets tricky with codominants. So these are codominant. This is recessive to these. And you can only get 2 alleles total, right? So you either get an A and an A, B and a B, an AB, an II. You can also get a combination, a little I, and you can get a B little I. If you get an A or a B combined with little I, you still have those enzymes. And if you still have those enzymes, you're still going to be a type A cell. Or if you have a B little I, you're going to have a type B cell. Okay? And that's where it gets confusing because not only do you have codominance here, but you have a dominant recessive relationship here. Good with that? I know that's a confusing one because we put it right under codominance.
I
there
understood.
any-
Go ahead, you'll carry what?
I said, I think you covered my question.
Good. Jen?
Are there any other examples of codominance? Because I can't think of anything outside of blood type.
I can't either, honestly. If I put something on the exam, it would definitely be blood type. There are other things I'm sure that are codominant. I don't know that I know one off the top of my head. I'd have to do some research.
Yeah, because I was running my mind there, and every example I can come up with would be the incomplete dominance.
Yeah, yep. I agree. I'm gonna look that up now that that's on my mind, if I put it on the right piece of paper here so I can find it.
Just for
Professor,
curiosity.
I'm gonna leave the meeting now.
Yep, go to your appointment. Bye, Carrie.
Thank you very much. Have a good day.
Thank you, you too. Okay. So are we good if we just practice some more Punnett squares, or did that bring up anything else that you need to have checked out along the way? All right. Then we can go back to this if we need to. I have a whole bunch of, let me pull this up here. I can find where I put it. Oop. That is not what I want. Where is it? Okay. So let's try some problems here. Let's see. Grab your piece of paper and you help me figure these out. So we have a mom who is homozygous for attached earlobes, which is a dominant gene. And she has a child with a man with unattached earlobes. What is the chance of having a child with unattached earlobes?
You said she was homozygous or hetero?
It says she's homozygous and attached earlobes. I thought unattached was dominant. I can't remember what example I used. This one I put that the attached earlobes are dominant. I don't remember in class what they were. Did I do them backwards? Hang on. Oh, for heaven's sakes. I can't believe how many slides we've covered this section. Okay. So unattached earlobes, is that dominant or recessive? No one can tell me that. I can't tell me that. Unattached earlobes is dominant. All right, to attached. So unattached earlobes is dominant. And mom has unattached earlobes. So we want to first determine our alleles. And I'm going to make U as a big U. Actually, you know what, you guys? Let's do this on the shared screen. I'm not sure how, this is actually a little easier as I recall, because there we go. All right, so let me make sure you guys can all write on this. Whoops, I've got to get out of this real quick first. Oh, wait, maybe I could do it here.
If we go to the top and we click viewing options and then annotate, we can edit on the screen.
Oh, you can without me having to make you a host? That's awesome. All right, so tell me what do you do? Or tell everybody else. Say it again.
You click viewing options at the top of the screen and then annotate. And then now you can, did you see my square?
Yeah.
Yep, we can do that now.
Okay. So mom is homozygous for attached earlobes. I was going to mess it up on here again too. And has a child with a man with unattached earlobes. What is the chance of having a child with unattached? So mom is homozygous for attached and dad has unattached.
What do we put for attached and unattached, like the letters and whatnot?
That's completely up to you. That's the catch with this.
Okay.
Is as long as you determine when you're doing the problem what it is, you know how to read the answer, right? And that's why I have you each time put the stuff over here, because, or on this side, and tell me what unattached and attached are for your definition. Because if you do that, then when you go to interpret it, you have what you used right there on the side. Homozygous or attached.
Mom is attached though, so.
I know my problem is bad because I had the unattached and attached backwards. But go ahead and let's see where we end up.
I had a question. So with we normally have like, so sorry. For mom, what was the two H words again? How to tell if it's, yes. What was it that again? Sorry.
I wrote the question wrong on my little sheet here. This is a sheet I've used before, and apparently I didn't notice it before. So let's switch this up. Because I don't think we'll be able to do it. Whoops, sorry, I'm writing on this. That's kind of cool, but I meant to erase, and I think I can erase. All right. We're going to go back and I'm going to restate this and we're going to make it right this time. Okay. So mom is homozygous for unattached earlobes. Let's get that one down. What is unattached versus attached? What do you want your unattached to be? Mom is homozygous for that. So pick your allele. All right, so UU is unattached. Then dad is attached. What do we know about dad? What do we have to know about dad if he's got unattached earlobe, or if he's got attached earlobes? He has to be two little a's, because in a dominant recessive relationship, right? In a dominant and recessive relationship, in order for him to express the recessive trait, he's got to have two of them, right? So we've got mom as unattached and dad as attached. Kind of odd alleles there, but let's go with it.
Will it work that they're different letters?
I find it gets confusing that way, but let's go with it and see what we find works.
If the mom wasn't homozygous, would that be, how would that change things?
All right, let's do this and then we'll change her to heterozygous.
Okay,
How's that?
sounds good.
And this is Sanvia. This is what you wanted to know, isn't it?
Yes.
All right. On this box, once you have given mom and dad their alleles, you can put, it doesn't matter whether you put dad's alleles on top or on the side. One of them is going to be mom's alleles and one of them is going to be dad's. And what this represents is dad can only, all right, let's look at dad here. Dad only has these two alleles. That means in all of dad's sperm, all of those gametes that he makes in every sperm, the only allele he can give his offspring is this or this. The same with mom. If mom is homozygous for unattached earlobes, the only allele she can give is this or this in the egg. She can't give anything she doesn't have. So that's where we get these. These are actually the if we imagine these as sperm and egg, these are what are going to be in the egg. This is what's going to be in the sperm. And then we go across this way with this and put it in each box. And then we go down this way and put this in each box. And this is giving us our combination at fertilization. If our big U and our little a, one from mom and one from dad, combine in our zygote, then we have this combination. If this U and this A combine, then we have this in our zygote. But we can also, now in this example, obviously these are all the same, but the principle is there that you're looking at the combination of gametes that can meet. And so you're going with this U, you're going down this way and putting the big U. With this A, you're going across this way and putting the A. You're doing the same thing here. All right, good. All right, so we have a bunch of U's and A's. What is that telling us?
All of the children are going to be heterozygous with unattached earlobes.
That is correct. I don't know about any of you, but the U and A would still be confusing for me. But as long as you put, what am I trying to do here? I'm not trying to erase. Let me try to draw. What the heck? I'm still erasing. Oh, okay. So if you put it over here though, as this is unattached, and this as attached, I suppose you could get it to work. Not conventional, but you're doing it for an exam. You're doing it on the side of your paper. As long as you can keep track, then you're fine. Okay, you can make those alleles anything you want. All right, now we were talking about what were we going to do? Make this heterozygous. So now let's take this out. Whoops, I didn't mean to take the whole thing. But let's now make mom heterozygous.
So does that mean that's 100% traits for the kids, right? Because those are all the same.
Exactly.
Okay.
All of them will have one allele, the dominant allele that they will express, but they will also have the recessive allele because of that combination. But I think we're going to see with this, because sorry, I got a little carried away. Go ahead. I'm not writing on it. I got a little carried away, but let's see what happens now, Sandhya. What was dad? Dad was attached earlobes. So he's still got to be little u, little u. So let's put in our Punnett square. And this time, I think we're going to find that it's a little bit different. This is where anything that looks the same in both letters, like I find that I'll do S, and yet S, big S and little s look the same. So you've got to be careful. Whenever I do a big S or I do a letter that looks the same as another, I do an underline for an uppercase. Just a little tip there. All right, so what's our Punnett square going to look like? Jen, you work on dad over here on the side. Katie, you do the top.
Deal.
Okay. So Sandhya, why don't you fill it in?
Oh, I honestly, I can tell you, but I just don't know how to work this thing that you guys are doing.
Oh, anybody
I
else got it working?
I can
Go
explain
ahead.
it again.
All right. Tell me what it is. Go ahead and tell me what this one should be.
Okay, so going across, those U's look like they're all lowercase. So
I know. You know what? But they don't have a line under them. So these are going to be lowercase. This is going to be lowercase, lowercase, and lowercase. This one has a line under it.
okay, all right. So we're gonna use uppercase U and then lowercase u, and uppercase U again and lowercase u. Those look like it's gonna be two little u's. And down there is gonna be two little u's.
Yep.
Yeah.
All right. So this time the difference with mom having two big U's and mom being heterozygous, having one of each, look at the difference in the offspring. 50% of the offspring are going to have dad's attached earlobes. Pretty cool, huh? All right, let's try a different one. This is a blood type one.
Just curious, sorry. Is
Go
there
ahead.
specific that determines whether it's homo or heterozygous?
Whatever I put on it.
I mean, like, so it just depends genetically what you have, whether
Yep.
they're not.
Yep.
Okay.
And since you aren't going to be able to see, and in a lot of cases I won't be able to see, the only time that you will know automatically is if it's the recessive trait and one parent has the recessive trait, then you know that they've got 2 recessive genes. But if phenotypically, if they have unattached earlobes, you don't know whether they have one dominant gene for unattached earlobes, or in other words, are they homozygous or heterozygous? And that's where I have to tell you in order to be able to do the problem. All right. A mother with blood type O has a child with a man who is type B. So we can put mom down, we can put part of dad. Their child is type O. This is one of those backwards ones. Their child is type O. What are the alleles for each parent? So dad is type B, so his phenotype is type B. So we may have to do 2 Punnett squares here, huh? What are dad's choices for alleles if he is type B? Are we stuck? There we go. So he can be B little I, right? Or he could be I with a B the capital B. That's the proper nomenclature. I don't care that you use it as long as you understand it. So what else could he be? What else could he be? What are his choices? If
Is
he's
there one with 2 B's, 2 capital B's?
Yep, exactly. All right, so we know that dad could be 2 capital B's, meaning he got 2 B alleles, or he could have a B and then his other allele is for O. So let's see if the one that's here is right, the BI. So let's put dad and mom's alleles up where they belong. There we go. All right. And so what have we got for results? And their first child is O. So it would appear that if dad's alleles are this, he could have a type O child, correct? What if dad is, let's change this spot right there. We go. Jen's on the ball. Let's now change him to be homozygous for the B allele. So he has BB instead of being BI. What does that do to his children? Is there any possible way he can have a type O child?
No.
No. So we know that dad's alleles in this case have to be BI.
Just curious, how do we know whether it's BB or BI?
Because Jen did this, and we would know because of the offspring. Because the question was, their child is type O.
So based off of what blood type the child is, depends on then how we tell what blood type the dad is.
Right, because this isn't possible to have a type O child, right? So if it's not possible here to have offspring that are type O, then we know that this cannot be dad's alleles, right? One of them has to be our little I. So that tells us we're working backwards.
Also, you would know that anytime you have two different homozygous parents, it's going to be 100% heterozygous.
Right. That is very true. Okay. I have a meeting coming up. Let's do one more. We can do one more quick. How about this is, Jen, you got good at erasing. Why don't you erase all the parts here? And if a carrier of cystic fibrosis, which is passed through an autosomal recessive allele, mates with another carrier of cystic fibrosis, what are the chances of having a child with the disease? You should know this from today, but let's work it out. So we have a carrier of cystic fibrosis. How do we want to name that allele? We don't need to put it on an X or a Y chromosome because it's not sex linked. If I said you have a sex linked disease, then you would associate it with the chromosome, but you don't have that. You just have any old autosome, just like you had your unattached earlobes and your type A and B. So we just need to name the allele somehow.
So it has to have a symbol that signifies it as, but it's not signified, it's not connected to our sex. So I'm blank. I don't know how to, do we pick any? Does it matter specifically?
No, it really doesn't. And that's why, remember, I always say write your alleles first. So is that Jen? Was that you trying to put something down?
Yeah, I was going to do little c because cystic fibrosis is recessive, right?
Yes, it is. How about, I have an idea. How about we do N for normal and little n for cystic fibrosis. Okay, that doesn't necessarily, that you don't have to do that, but that is two obvious letters. I can make this normal and this CF. Okay, so hopefully we'll go with something like that. So if a carrier of cystic fibrosis mates with another carrier of cystic fibrosis, so what are our alleles for both of them?
Big N, little n.
Big N, little n. They're both carriers. That means they both have the bad allele, but they also, remember that this is a recessive allele, right? So it's in small. In order for it to be expressed, we have to have two recessives. So both of our people are big N, little n, big N, little n. And so we end up with how many carriers do we have of cystic fibrosis?
1.
2. 2
What is it?
carriers and 1 with cystic fibrosis.
Right. So this person got both recessive genes, right? Both recessive traits, and they're going to, they just happen to be cystic fibrosis, unfortunately, right? It's not attached earlobes. So they've got both abnormal genes, so they have CF. And then these two will be carriers of the gene, but will not have CF. And then this will be unaffected and not be a carrier.
Will we ever have a problem where we'll start with Lowercase, lowercase.
If you have a homozygous recessive presentation, you will.
So, and that would, instead of it being capital, they would both be lowercase.
Um,
Like the way we've set it up now.
are you talking about if we had cystic fibrosis, would we ever have a little n to start this?
Yes.
Yes, you could. You could. I mean, people with cystic fibrosis do have children. Usually they can live relatively normal lifespan. So we could have this be, well, we could have, there we go. Somebody get rid of that for me. Where are the bees coming from? Oh, jeez. All right. So now, all right. Let me try. All right. So I'm going to now have one of the parents have cystic fibrosis. So they're going to have little m. And if they marry someone who has or is a carrier, let's make them be a carrier, right? And so we're going to have n. We're going to have, there's one that has cystic fibrosis. And there's another that has cystic fibrosis. So 2 of their 4 children have the probability of having cystic fibrosis. Each child has a 1 in 2 or 50% chance. These 2 would both be carriers. So it just depends on how we start and what I use as a word problem, Katie, and where your starting point would be. I do want to do one more. Let me see. I should have done an incomplete dominance. Let's see, though, if I have I think I have hemophilia is a sex-linked disease. So is Jen cleaning that all out for me? Awesome. So if hemophilia is a sex-linked disease, whenever I put that it's a sex-linked disease, or I'll probably be more specific and I will say it's an X-linked disease. So if I have hemophilia, and what's my question here? What is the likelihood of having a child with hemophilia if mom is a carrier and dad has the disease? So the first thing I want to do is I want to show my normal alleles, X and Y. So those are my normal alleles. Hemophilia is X-linked. So I want to make sure that I put it on my X chromosome. So this is my abnormal allele. My mom is, what did I say? Mom is a carrier. So that means mom, first of all, has 2 X chromosomes, right? But if she's a carrier, that means she has 1 of the trait, the hemophilia trait. So 1 allele is abnormal. Dad has hemophilia. And since he is XY, then the only X that can have hemophilia is his 1X. So when I get done and get ready to set up my Punnett square, I have to have mom and dad's alleles, and I have to have them with the X and Y. So there it is, mom and dad's alleles. So I have mom, she has an X, and then she has an X that carries the bad gene. And then dad has hemophilia, so his only X gene is bad, but his Y gene is fine. So we have X, H, X, H, X, H, X and Y, and then X, H, and Y. So first of all, we know that there are 2 boys, which makes sense, right? 50% are likely to be boys. So we have 2 boys. We also have 2 girls. The boys, first of all, this one didn't get any abnormal gene. So that person has no hemophilia. He's perfectly normal and has normal genes. This person only has one X allele, and that is abnormal. He will have hemophilia because his only X allele is bad. Well, let's start here. This person, this female, is a carrier, right? She has one good and one bad gene. She will carry hemophilia like her mother, but she will not have hemophilia. And then we have this daughter, and this daughter is going to have hemophilia because she inherited both X alleles that have the bad gene. So she will be a hemophiliac, he will be a hemophiliac, and she will be a carrier. Okay, so when I say X-linked, the first thing you have to do is to determine your alleles and then make sure that you attach the abnormal allele to an X chromosome. Okay, it's going to be wrong if you don't, if I ask you to do the problem and you don't attach it to an X chromosome, then it's going to be wrong. Okay, so if you're doing it in front of me or I have you fill it in, I haven't decided yet how to do it. With an X-linked, you've got to make sure that you attach it to your X chromosome. All right, and I have to go now, but any quick questions?
I did have a quick question. I forgot to email you my test extension yesterday. Can I still email it to you today?
Your test extension.
Basically, what is it called? Accommodations.
Oh, yeah, go ahead.
Thank you.
All right, have a good day.
Thank you, you too.
I don't think I have any questions
All right,
right now.
all right. Good luck studying. I'll see you later,
Thanks.
Dylan.
Yep. You too.
Bye-bye.